Hello, everyone, and welcome to today's webinar titled Pharmacogenomics, Post-COVID-19 Expansion of Molecular Testing. This webinar is part of the 13th Annual Clinical Diagnostic and Research Virtual Event. I am Megan Pascal of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, go to www.thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate with communicating with other attendees using the new live chat feature during the presentation. You can find the live chat located on the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. You will notice that the speaker has provided a survey located to the right of the Ask a Question box. This survey will be available during both the live session and the on-demand period. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. I now present today's speakers. Dr. Michael Donovan, lecturer and biomedical diagnostics at Arizona State University. Mara Aspinall, managing director, Health Catalyst Group and professor of practice, biomedical diagnostics, Arizona State University. And Dr. Marilyn Owens, founder and principal at Medical Laboratories Strategy and Leadership, LLC. For a complete biography of our speakers, please visit the Presenter tab from the menu at the left of your screen. Dr. Donovan, Ms. Aspinall, and Dr. Owens, you may now begin your presentation.
My name is Mara Aspinall, and I'm very pleased to welcome you to this session. My focus in life really is diagnostics. I've spent the last 30 years in the industry as CEO of Ventana Medical Systems, now Roche Tissue Diagnostics, and president of Genzyme Genetics. But I'm most proud of being the co-founder and professor of practice at Arizona State University's program in biomedical diagnostics, focused on teaching diagnostics for the existing industry and the future. But today is about both of those, the existing industry and the future. And we have the privilege of hearing from two great speakers who are leaders in the diagnostics industry, Marilyn Owens and Michael Donovan. Marilyn is a senior healthcare executive with over 40 years of extensive management experience establishing and leading clinical reference laboratories, including academic medical centers as well as private and hospital labs. She has a broad scientific background in immunology, infectious disease, oncology, genetics, and molecular biology. Marilyn has substantial lab operating experience worldwide, including P&L responsibility with both small and large labs. Her expertise spans startups, M&A, and strategic planning. Marilyn is an experienced teacher, mentor, and coach with graduate training in business as well as life sciences. Our second speaker is Michael Donovan. Michael comes from a very different background. He is a registered patent attorney specializing in patent prosecution and intellectual property licensing and commercialization for life science-related technologies. He has an academic background in microbiology as well as a PhD in immunology. After leaving private practice, Michael worked as the intellectual property counsel for the Translational Genomics Research Institute, and most recently served as system director for intellectual property at Common Spirit Health. Today, Michael is a lecturer in the ASU Biomedical Diagnostics program, where he teaches a variety of areas, including regulatory law, health economics, and bioethics. As we've discussed, we're going to talk about pharmacogenomics in the post-COVID-19 expansion of molecular testing. Today's agenda will focus on 4 topics. First, changing expectations for COVID-19 testing. As we hope to be in the last stage of the pandemic, we all know and have seen that the expectations for labs and lab testing has changed. We will talk about those changing expectations and how they affect your lab. Secondly, the evolution of consumer expectations, not just for COVID-19, but how consumers are thinking about testing more broadly. Third, what are those opportunities post-COVID for your lab? And in particular, we will focus on pharmacogenomics. And lastly, how is the best way to repurpose your lab assets for growth in the future around pharmacogenomics and potentially other areas? So we look forward to an active presentation and, as Megan discussed earlier, answering your questions at the end. Over to you, Marilyn.
Thank you, Mara. The COVID-19 pandemic has dramatically changed the way clinical laboratories operate. And I think all of you recognize that our lives will never be the same again. Molecular diagnostics are really critical in detection of these nucleic acids and proteins, not only infectious disease, but cancer and genetics as well. And this is up to the laboratory to perform the appropriate tests for physicians and patients. As a result of the widespread infections of COVID-19, only molecular techniques could meet the standard of speed, sensitivity, and high volume testing. Most labs ended up employing multiple testing platforms, partially because of supply chain issues, and rotated staffing to cover 24/7, increasing customer service for collection, processing, and reporting. The evolution of COVID-19 testing in the EU started with the global initiative on sharing avian influenza data and enabled nucleic acid amplification tests to be diagnostic for the SARS-CoV-2. The RT-PCR became the cornerstone of testing in Europe, as it was in most other countries, with the use of other molecular techniques becoming innovative for transcription-mediated amplification, the TMA or CRISPR techniques. Time was of the essence. I can't say that more emphatically. We had to streamline collection centers, automate workflow, and generate results almost as quickly as that they could come off of the instrumentation. As of July 22, there were 418 RT-PCR assays which had received CE marking by the EU Commission. Laboratory relationships and customer relationships all changed. We had to really scatter to be able to handle the response that the community and the corporations, public institutions, travelers, all of the demands on individuals and their proof of having been tested for COVID. And the focus was directed to be as convenient as possible for consumers, closer to their homes, testing around the clock so that they could get there around their working hours, clear and concise reports so they knew what that meant, and electronic and rapid reports provided not only to the physicians, but to the patients themselves. After RT-PCR became the standard, and it was taking time to do the tests, to do them accurately, the lateral flow antigen tests became popular, especially by those who prefer the convenience and privacy of doing their own testing at home. Consumers want to manage their own health. The changes in consumer expectations, I think, just went hand in hand with the COVID epidemic. They want to take control of their health. They want to order their own tests, study their test results, and monitor their health with digital devices. Everybody has a smartphone. Everybody has a digital device that they could use to monitor. We learned from the COVID pandemic that today's consumers want access to convenient healthcare. Home collection dominates collections at local pharmacies, clinics, or laboratory patient service centers. And while consumers still want to discuss their health with a healthcare provider, they're amenable to virtual visits. There has been a huge boom in virtual health, and it's primarily because of the COVID-19 pandemic. Most of all, consumers want to have their diagnostic information at their fingertips when they want it and at a reasonable cost. As I said before, time is of the essence. It's no surprise that consumers want their results quickly and accurately. With the improved access that has come from convenient testing locations and at-home testing, it's really expanded the opportunities for access to care. And this quest for health equity has really been given a jump start because of the COVID pandemic. Underserved communities now are getting more care. The opportunities for the post-COVID clinical lab are significant. If you just think about the volumes that we have been churning out over the past year, June of 21, there were approximately 170 million PCR tests that had been performed, whereas last June, it's down to about 70. So clearly, we don't have the same volume constraints, and we don't have the need for all of the staffing, collection equipment, and testing equipment. As these volumes continue to decrease, we don't think they'll ever go away. But there are opportunities to repurpose the assets that were put into place to handle the pandemic. The first option would be to repackage COVID testing with other respiratory pathogens. And a lot of vendors have already done this. So molecular panels, which include COVID, might also include flu A and B, and perhaps RSV. Secondly, combat the great resignation of the loss of laboratory trained individuals by deploying personnel to cover other departments. They've been staffed up for COVID, and we can reuse those individuals in microchemistry, other departments, as well as other shifts. Thirdly, the consolidation and strategically focused laboratory acquisitions are now available. So if the laboratory is looking for an exit strategy, there is a lot more opportunity for that to happen at this time, because they've expanded, they've made money off of COVID, and if they want to retire or consolidate, there are people who are willing to do that. But fourthly, these opportunities are opening up more laboratory options for pharmacogenomics. As Mara said, it's the topic of this seminar. Next, Michael is going to continue to describe pharmacogenomics.
Thank you, Marilyn. Now we're going to move on to a little bit of pharmacogenomics background. So pharmacogenomics, or as we'll refer to it sometimes as PGX, plays an important role in identifying responders and non-responders to medications. You can think of pharmacogenomics as a subfield of genomics in which the particular patient's genetic profile not only impacts how they metabolize a drug, but also what potential side effects they might suffer from. So if you think of kind of the old paradigm where a particular patient group, let's say those who are suffering from type 2 diabetes, receive a drug, now within that patient group, there are different subpopulations. So for some, the drug might be non-beneficial, meaning it might not work, and it might actually be metabolized into a toxic byproduct, in which case you definitely don't want to take it. As we move around this circle, though, you see in some cases the drug might be non-beneficial and non-toxic, in which case you still don't want to take it. Next, you might see, going around the circle, you might see a drug that is beneficial but toxic, in which case both the patient and the healthcare provider will need to have a discussion about the potential impact of that drug on that patient's quality of life. And finally, you get the relationship that you really want, which is a drug that works, it's beneficial, and it's non-toxic. So commonly, some of these genes are often associated with the cytochrome P450 group. These are drug metabolizing enzymes, although there are other pharmacogenomic biomarkers that do play a role as well. So next, let's talk a little bit about how this current paradigm, like I just mentioned, is growing to be unacceptable and potentially unnecessary of just prescribing a medication in a one-size-fits-all context. For example, to be clear, most medications don't work for everyone. When you look at the top-selling medications that are out there, 90% of those medications only work in 30 to 50% of the population. You have a lot, that means that there are a lot of prescriptions that are being written for individuals who are not actually seeing benefits. And if you look in on the right here, you see that things like diabetes, arthritis, Alzheimer's, in all of those cases, at best, you're seeing a 57% success rate. So the current paradigm is leaving a lot to be desired in terms of both economic loss in terms of paying for drugs that aren't working, as well as potential loss of chance and physical harm to individuals who are suffering from adverse drug reactions. So now I don't want to short sell prescription drugs. Obviously, they've had a tremendous impact on the modern practice of medicine. So there is most definitely benefit to them. But as we've just talked about, there's some drawbacks. Now, when you consider that 82% of American adults take at least one medication, that is a huge group of individuals who have the potential to suffer what are called adverse drug reactions, or basically these are responses by an individual that might lead to negative health consequences. So adverse drug reactions can lead to a waste of time in terms of opportunity for proper treatment. They can add cost in terms of additional treatment that needs to be sought out, and they can lead to increased risk to the health of that individual. As a result of these, you can see millions of additional visits to not only the emergency department, but also the doctor's offices and the costs associated with that. But you can also see a significant impact on morbidity and mortality and the associated economic impact on these healthcare systems. So for the last few slides, we've talked about pharmacogenomics and the importance that it can have. When we look at these additional numbers, 90% of individuals have a genetic variant in a drug metabolizing enzyme that if they're given a drug that is actually metabolized by that enzyme, you could see varying responses. And then combine that with the fact that 35% of Americans over the age of 70 use 5 or more. We just talked about how 80% take at least one medication. This is 5 or more medications. So there is a significant number of, at the very least, Americans, and on a global perspective, even more people that are at risk for adverse drug reactions. And given that, pharmacogenomics is an opportunity to use either that underutilized or unused space, equipment, and personnel that are no longer immediately needed for the deluge of COVID-19 testing. Now, we are not advocating that you completely scrap your basic testing capabilities for COVID-19. The fall and the winter are just around the corner, which means that there could be another surge. So you do want to maintain some basic testing capability for COVID-19 that can be used. But deploying some of those unused assets does allow you to potentially take advantage of this pharmacogenomics movement. Now, from a pure numbers perspective, the pharmacogenomics market from a global perspective is growing. This year, it's estimated to be a little bit over $13 billion for pharmacogenomics testing, but over the rest of this decade, there's expected to be a greater than 11% combined annual growth rate up until 2030, where you're expected to see $33 billion in annual testing revenue. Now, a laboratory wouldn't necessarily need to completely retool In fact, the PCR segment of this pharmacogenomics testing market is actually the largest technology used, with about 40% of the overall market in 2021. So there is not only significant revenue to be had, but also it can still be based on conventional molecular diagnostics. Now, there is broad utility for pharmacogenomics. Obviously, most people know about the impact of pharmacogenomics in oncology. HER2 and the use of Herceptin is one of the early successes of pharmacogenomics, but there are other areas as well that where pharmacogenomics plays an important role, such as cardiology, where the use of drugs like Plavix and warfarin are definitely known to there's variability in the way those drugs are metabolized based on the genetic background. Similarly, with pain management and perioperative and postoperative care, where you see challenges with respect to the way an individual might metabolize an analgesic or an anesthetic. And then finally, there's also mental health. And mental health is obviously becoming such a huge, important area. But unfortunately, for some mental health related drugs like SSRIs, you see a 2 week to a month lag before those drugs reach therapeutic levels. Think about the opportunity loss there when a healthcare provider doesn't necessarily know if that person is going to metabolize that drug properly. If they know that in advance, they have the opportunity to avoid that drug and make good use of that 2 week to 1 month time period on another drug that might work better. Now, not only are these areas where you could have utility in pharmacogenomics, these are also areas where laboratories who have service lines in these areas can also obtain samples. And as we're going to talk about in a little bit, having proper verification and validation of your assays is going to be important for obtaining reimbursement. So reimbursement, as I just mentioned, is an important question when it comes to any type of healthcare-related service. Long story short, the reimbursement system in the United States is evolving and extremely complicated. For those of you outside the United States, there are basically two large categories. There's CMS, Centers for Medicare and Medicaid Services, government-funded healthcare system or healthcare payers. And then there's third-party payers, private independent insurance companies who pay medical expenses for a large number of Americans. The challenge here, though, is that for CMS, that government-funded healthcare, there are very high-level requirements in order to actually get coverage for pharmacogenomics. So the results must be considered reasonable and necessary for a specific patient. There has to be a reasonable and necessary reason to conduct this assay. You also need to have clinically actionable data that's been reviewed by the FDA in order for that particular gene-drug interaction. So basically, you need to have not only a specific reason, and it's got to be necessary, but you also need to have that background in terms of that clinically actionable data. Now, there are important limitations, such as you need to have established analytical and clinical validity as well as clinical utility. And this can be a challenge for some laboratory developed tests because you don't necessarily need clinical utility for those, at least to get past CLIA regulations. So it might require additional effort. And then also, if you are undergoing germline testing, you're only going to get that paid for once in your lifetime for a particular gene. Now, how can you actually establish coverage? Through either a national coverage determination, which covers the entire United States, or a local coverage determination, in which case a Medicare Administrative Coordinator or Contractor, or a MAC, will actually make a determination for a particular test in a particular geographic jurisdiction. So kind of you have the ability to look at things either nationally or within a particular region. And then in contrast to what we just described with respect to the government, there are also third-party payers. These are those independent insurance, or these are these non-governmental insurance companies, and you can have varying coverage depending on what information that insurance company actually wants before they're going to decide to pay for pharmacogenomic testing. So how do we get some of this information? Well, the pharmacogenomics guidelines are actually able to define the value proposition here. And in particular, there's a consortium called CPIC, or Clinical Pharmacogenomics Implementation Consortium, which is an NIH-funded organization of a variety of different stakeholders from academia, such as Stanford and Vanderbilt, as well as nonprofit organizations like St. Jude's, clinical labs like Arup, as well as healthcare systems like Geisinger. They all have come together in order to advance the cause of pharmacogenomics. They publish guidelines and grading of gene-drug groupings. So basically, they have set out a scale of different gene-drug interactions in terms of whether or not those interactions should be looked at before prescribing a particular drug or a particular drug at a given dose, in which case you're at level A, all the way down to the bottom at level D, where there might be some published studies, but the clinical actions associated coming out of those studies aren't particularly clear, or there isn't sufficient clinical data yet. So CPIC is an advocacy group that is really pushing to get this data out there for use by clinicians, laboratories, and potentially patients to understand their own healthcare. So outside of the US, what does the reimbursement situation look like? So unfortunately, pharmacogenomics is rarely covered and reimbursed by healthcare insurance in a variety of countries outside of the US. In fact, the awareness and understanding of pharmacogenomics is extremely low in low and middle income countries. And a lot of this is tied in to the US's inconsistent regulatory and reimbursement landscape impacting these other countries. So they will look to the US in terms of what is the US government paying for. And as I just mentioned a few slides ago, it's questionable. It's kind of, in some cases, it's on an ad hoc basis. For example, you'll see some adverse drug reactions being covered or a lack of response to medication. You might see coverage for pain management, cancer management, as well as comorbid conditions. So you do see some inconsistencies, which kind of has a domino effect across the globe. However, in the EU, there's a group called the Ubiquitous Pharmacogenomics, or UPGX, which is much easier to say. Similar to CPIC, they provide studies and education for healthcare providers to increase awareness of pharmacogenomics. So they are pushing pharmacogenomics just like CPIC pushes pharmacogenomics here in the US. They document effective treatments for European citizens, and their advocacy includes things like cost utility analysis, as well as looking at direct cost savings that should lead to coverage of these pharmacogenomic techniques. So how could a lab or other healthcare organization get into pharmacogenomics and potentially repurpose some of its assets? One is getting involved in clinical trials. So, for example, we have, this is from an article about what Merck is doing in their clinical testing. Merck, for the last few years, has actually taken genetic samples from a lot of their clinical trial participants and holds on to those for kind of downstream usage. So, for example, at phase 1, they'll obtain these samples. Phase 2, they could do something like a GWAS study or whole exome sequencing, genetic profiling, to basically try to understand then for phase 3 whether or not you can create some type of stratified trial, or if you're seeing a large disparity in response to a given drug. Basically, this data has the ability to go out, or to be used to go out and create a great cohort for phase 3 studies. So you are looking at you have a much more better likelihood of a positive result coming out of your trials. So obviously, in addition to that, obviously this is another great source of potential samples that could be used in your in-house assays. Now, pharmacogenomics, although there is a push here, we're having this conversation, there is a certain maturity. This is not a new technology by medical standards. It's been around for a few decades. Pharmacogenomic biomarkers in particular This study looked at the prevalence and the presence of pharmacogenomic biomarkers in FDA-approved drug labels for the first 2 decades of this century. And here's what they found. They found, not surprisingly, oncology is still the primary driver of these pharmacogenomic biomarkers and drug labels, but there's a bunch of other medical specialties as well, like dental, anesthesiology, pain management, neurology, mental health. Again, all these different areas are starting to recognize what the importance is of having that pharmacogenomic data when making prescribing decisions. Moreover, when you consider the 250 drug labels that came out that had pharmacogenomic data, a lot of those labels, 94 of them in fact, actually required pharmacogenomic testing before prescription. There were also some that suggested it, where it might be actionable, and then there's also a proportion similar to those that were required, where it just provides informative data. But this shows not only are we seeing the presence of this data present in these drug labels, but it is making it, the FDA is requiring action in a lot of circumstances. So up next, we actually have a study that was a collaboration between Cornell Life Sciences and the Teachers Retirement System of the state of Kentucky. This is a great example of where a pharmacogenomics, an all-encompassing holistic plan can make a significant difference. So the Teachers Retirement System of Kentucky is composed of retired teachers. And in this study, 10,000 plus retired teachers have enrolled in it. And what this study is a combination of education on pharmaceutical management and as well as genetics and pharmacogenomics, collecting of samples and genetic testing to figure out what genotypes these individuals have, and then medication management, where a pharmacy service within the state of Kentucky took that clinically actionable data and provided recommendations to these enrollees. Overall, it was found that 87%, almost 90% of the enrollees were at high risk of drug interaction. And 3 out of 5 of the retirees had recommended changes to their medication strategy. Now, this is an improvement of the potential health impacts on these individuals. But if you look over here to the right, you also see, most importantly at the bottom here, over the course of a 32-month study, so that's 2.5 years, a savings of $37 million. That is not only cost avoidance, but also savings in terms of potentially unusable drugs that are potentially harmful and or non-beneficial drugs. So you do see not only the clinical benefit, but also the benefit to the healthcare system as well. And then finally, from an example perspective, we have a potentially impactful deployment in the European Union. This is an economic analysis. This is, just to be clear, this is not this is not the same implementation that we talked about on the previous slide in Kentucky, but this is what is thought would happen if a similar system was implemented in Glasgow. So Glasgow has, in terms of those who are 65 plus in Glasgow, the average number of medications that are being taken is 8 medications per day by those 65 or older in Glasgow. And if they were to employ a similar system in Glasgow, the estimated cost savings is over 85 million pounds over the course of 3 years. And again, this is not a real result. This is a model based on what we saw in Kentucky in terms of implementing that same system. But it does show that there is significant benefits out there to be had simply based on the implementation of a little education, some genetic testing, as well as medication management. So overall, what we've talked about in today's seminar is how the COVID-19 pandemic was responsible for laboratories making some huge changes. They faced a tsunami of molecular samples and came to grips with those challenges. And we learned as an industry how we can respond to unexpected events. But now that COVID is slowing down, hopefully for the long term, there comes an opportunity for laboratories out there to make use of this extra bandwidth. And pharmacogenomics has tremendous potential to make an economically beneficial impact on so many individuals that are out there. And again, remember these numbers. There's 5 billion prescriptions written every year, and 82% of American adults take at least one medication. On the flip side, we have adverse drug reactions with 3 million people suffering from them each year, which makes it the 4th leading cause of death. When you think about it, pharmacogenomics has the ability to make an impact in different areas of medicine like behavioral health, cardiology, oncology, and what have you. But more importantly, it has the ability to make an impact not only on the healthcare outcomes of individuals, patients across the world, but also has the ability to help from an economic and a healthcare planning and management perspective.
Thank you, Dr. Donovan, Ms. Aspinall, and Dr. Owens for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsors, Thermo Fisher Scientific, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until November 9th, 2024. Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.